Any questions before we get started today? Alrighty. I'm trying to record this for a future hybrid class in the spring, so we'll see how it goes today. I have some terrible jokes. What do you get when you cross a centipede with a turkey? Drumsticks for everyone. What do you call a six-legged insect that cannot speak? A mute ant. Having sex in an elevator is wrong on so many levels. How many tickles does it get to get a squid to laugh? 10. 10 tickles. Tentacles. Probably one of my all-time favorites. A pig walked into a bar and ordered 15 beers and promptly drank them all. The bartender asked, "Would you like to know where the bathroom is?" "No," said the little pig. "I'm the little pig that goes wee, wee, wee all the way home." Alrighty. Okay. We're going to talk about proteins today, one of my favorite groups. And as I mentioned last time, proteins are really the machinery of life. If you really want to see what the structure of a cell is, or an organism, what runs the cells, what maintains the cells, or what recycles the cells, you're going to see that most of it's going to be protein. And last time we were talking about how important the shape of a protein is in their functions. Today I want to talk a little bit about a fantastic technique that was developed just over 100 years ago as a way to figure out the structure of a lot of molecules. And this was developed by a father and son team called the Braggs. And we've used it for 100 years to decipher the structure of all kinds of things. And one of the big ones are proteins. And I showed you this little picture here. It kind of looks like a big party favor. But this represents a really common protein that we find on all of our skeletal muscles. It's a little receptor for the neurotransmitter acetylcholine. Now, how we actually go and figure out something like this is to first start with some crystals. The process I'm going to show you is called X-ray diffraction crystallography. All right. Now you know that if you go and you take, let's say you have a solution of water, let's say you've got some salt water, and you let it sit, and as the water evaporates, all of a sudden you get this precipitate down at the bottom, and eventually when all the water goes away, you're left with these little salt crystals. Well, it turns out, salt isn't the only substance that, given the right conditions, will turn into a crystal. Right? Crystals are wonderful in a sense that they have an ordered sequence. Every atom of every molecule is going to be arranged in the same order. So all of a sudden you have this repeating pattern of all these similar molecules, all with the same orientation and same structure. So for a chemist, that's a great way to sort of start out. So what we do is just like we do with salt water, we can do it with proteins. So we can take an unknown protein, put it in water, all right? There's a little bit of tricks to it. You'll show, and I'll see in the little film that there are a couple little tricks to this. All right. As the water evaporates away, eventually these proteins, just like salt water, will precipitate out into these crystals. Here's a couple of examples. This one here is crystals of a protein called lysozyme. Lysozyme is a protein that you find in your tears and your nasal secretions. Its big job is to break down the cell walls of bacteria. So it's an antimicrobial or an antibacterial agent that our cells make. So here we have some crystals. We put it in a little box of crystals. So we have some crystals. Now, turns out that oftentimes when we use microscopes, we're relying on light. Well, light comes in different wavelengths. Visible light has a certain wavelength, but the structures that we're going to be seeing are things that are smaller, that can be smaller than detected with visible light. So we need a really, really short form of light to get into all the nooks and crannies and reflect back. And so in that case, we use one of our short forms of light, and that's an X-ray. X-rays have much, much shorter wavelengths. That way, they can actually go and be blocked or diffracted. And what we mean by diffracted, bent. All right? So those wavelengths of light at the X-ray can be bent by the atoms that are there in the crystal. And you'll notice that when we do that, it produces what we call a diffraction pattern. Every element, every atom of every element, all right, has a certain number of electrons. Okay? And it turns out that those electrons can bend those X-rays. So the greater the number of electrons there, the more bent it's going to be. The fewer number of electrons, the less it's going to be bent. So all of a sudden you have a way to identify different elements by how much the light is diffracted, how much the light is bent. Right? And so we come up with something that looks a little weird, a whole bunch of dots. Right? That's a diffraction pattern. We can take that and put that in a little algorithm, and it will translate that geometry into what the molecule looks like. Now, the cool thing about it is it'll do it like an MRI. If you've ever seen an MRI do it, we do it in a series of slices. Same thing happens with a crystal. We'll have a band of X-rays go through one plane, and then another plane, and another plane, and each one will produce its own little diffraction pattern, and we put those together to make a three-dimensional molecule. And most proteins, the structures are figured out this way. Now, let me show you a little video that shows the process. Now it's there. Go to my documents. The video.
If you want to determine the structure of a protein by X-ray crystallography, you first have to grow a protein crystal. Unlike table salt, proteins don't crystallize easily because of their irregular lumpy surfaces, so numerous attempts may have to be made to make a crystal. Various factors, pH, temperature, the solvent, are important for success. Salting out agents such as ammonium sulfate can reduce the solubility of the protein to aid crystallization, but it may take several days to produce a crystal large enough to use. The protein crystal is mounted in a specific orientation and exposed to a beam of X-rays. recorded on film. Each atom in the sample scatters X-rays, and the strength of scattering is proportional to the number of electrons in the atom, so each atom can eventually be identified. Since you can't focus X-rays with a lens, a different method is used, a mathematical formula called a Fourier transform, which can interpret the X-ray photograph to produce a contour map of electron density. This map represents the electron density for a particular plane through the crystal. The peaks and valleys correspond to the positions of atoms. Here's the heme in hemoglobin, whose atoms all lie in the same plane. The hill in the middle is an iron atom. Since proteins are three-dimensional, many such maps, each representing a slice through the molecule, must be assembled by computer to create a complete 3D structure.
Alrighty. Wonderful. Now, this process, as I mentioned before, has been in use for over 100 years for all kinds of different molecules. You can imagine that some of the early ones were very, very simple. Our first protein that was actually deciphered this way is one that we find in our muscle. I've just bought my wife a couple of ribeye steaks yesterday. She's a big steak lover, and of course my German Shepherd is also a big steak lover too. And if you've ever wondered what the red color is that is found in beef, it is rich in myoglobin. It's actually, that's the molecule. And people often are saying, oh, that's blood. It's not blood. That myoglobin is there as a way to store oxygen. It's very similar to the hemoglobin that we find in red blood cells, and since your muscles require a tremendous amount of oxygen, that's where a lot of the O2 is stored in the myoglobin. And this was the first protein to be deciphered using X-ray diffraction crystallography back in the mid-20th century. Now let me show you a couple little pictures of different types of proteins. And I'll put in my... There we go. Now, here's a nice little picture of what we call a space-filling model. You can see each one of these little balls represents an amino acid. But there is a different type of model, and we saw that with the acetylcholine receptor, one that looks like a ribbon, and this is called a ribbon model. Both are useful for our purposes. We'll see that the ribbon model works the best for us, but for chemists and for biochemists, both of them have their place. But this is what lysozyme looks like. We saw the crystals of lysozyme, but this is what the actual protein looks like. A few others just to show you some cool ones. A lot of hormones are proteins, and here are a couple of them. This one is prolactin. This is produced by women in their last trimester. And it actually goes and signals the breast tissue to develop and start making milk. And if you look at the name, IN tells us it's a protein, and this is for lacto, meaning milk. This is another hormone. This is a hormone that's secreted by your thyroid gland called calcitonin. We used to know what it did, but our hypothesis about what it did has been falsified. So it's now a hormone in search of a function. So hopefully it'll be interesting to find out what exactly it does. This is a defensive protein called an antibody. Our little B cells go and form that. This is trypsin, which is a beautiful little enzyme that breaks down proteins. And this is one, if you're a fan of endurance sports, if you look at particularly cycling or cross-country skiing, this is the one that busted Lance Armstrong. This is actually a hormone that is produced by your kidneys, and it stimulates your bone marrow to make red blood cells, so it raises your red blood cell count. Red blood cells, since they carry oxygen, especially if you think about an endurance event like cycling or marathon or stuff like that, you can see why that'd be something that you may want to play with by increasing your EPO and carry more oxygen and therefore increase your endurance. So that's a big target for the anti-doping agencies. All kinds of venoms. In fact, one of the things I'm really fascinated about, just as a biologist, is venom. And we know there are a lot of organisms that have venom. If we think about jellies and corals, and particularly we look at arthropods, scorpions, and insects, and all kinds of things like that. We even have venom showing up in reptiles, and of course we have a mammal with some venom. If you didn't realize it, male duckbill platypus actually can inject venom. They have a very painful little spur on their hind leg that they can cause you some serious pain. But venoms, believe it or not, are mostly going to be proteins. What was that? Good question. When we start talking about the structure of the proteins, we'll see that our little spirals, these are one type of structure called an alpha helix. And then the arrows that you're seeing there is another type of structure called a pleated sheet or a beta sheet. Oftentimes, just like diseases or genes are often named for ones that don't work right. I'll give you a little example. There's a mutation that occurs in fruit flies where the fruit flies are born without eyes, no eyes whatsoever in the adult. So they've located the gene that's responsible for that, the gene that doesn't work right, and they call that the eyeless gene. All right? So genes and proteins are often named for versions of those that don't work right. This how do we sort of discover what they are and what their function is. Turns out that we have an important little gene that is involved in making a muscle protein. This muscle protein is called dystrophin because it's malformed in people with muscular dystrophy. This actually goes and helps hold the little muscle fibers to the cell membranes that we have in skeletal muscle and smooth muscle. And it's a gorgeous, gorgeous protein when it's working right. If you're a fan of glow-in-the-dark fish, you can go to Walmart and buy yourself a fish that glows under ultraviolet light. That glow comes from a beautiful protein that's found in jellyfish. This is green fluorescent protein. We discovered a gene for it in a jellyfish, took the gene out and started putting it in all kinds of organisms, including, I've seen green fluorescent pigs, rats, mice, monkeys, rabbits. What was that? Oh, that's genetic engineering. We did it. Yes, it's been modified. It turns out this has some really cool applications. If we go and we attach it to certain parts of the cell, for example, if we attach green fluorescent proteins so that they're expressed with spindle fibers during cell division. Then we can watch the spindle fibers. It's a great way to actually go and see what's going on inside a living cell. So, pretty cool. Now, one important hormone that's also a protein is insulin, and this is just showing you the 3 types of models. You can see our ribbon model. This is our space-filling model. And this one is kind of complicated. This is a ball and stick. You might remember the ball and stick model that we learned on the first day with water. Now, proteins are really just kind of like strings of pearls, like a pearl necklace. They're just a long polymer. Some of these are huge. Dystrophin, for example, has over 100,000 amino acids in a chain. So some of these can be ginormous. But if we think about a pearl necklace, all right, the necklace itself is the polymer. We call that the polypeptide. We'll find out that there are some proteins that are built from more than one polypeptide. And the pearls themselves are what we call the amino acids. Now, let me show you what insulin looks like. We saw how it was all folded up, but this is what it looks like when it's unfolded. Turns out insulin is composed of 2 chains, 2 polypeptides. This is our alpha chain, and this is our beta chain. And if you look at it, you'll notice that every little amino acid, we often abbreviate it by the first 3 letters. For example, this is glycine, isoleucine, valine, glutamic acid, glutamine, cysteine, threonine, and so on and so forth. One of my favorite parts of this is you'll notice a strange little bar coming off with an S here and an S here. And they always come off the cysteines. Turns out what holds a lot of proteins together and help define their shape is a type of covalent bond that's unique to proteins called a disulfide bridge. So when we have a cysteine amino acid bonding to another cysteine amino acid, turns out that they're held together by what we call a disulfide bridge. And as you can see, it can go from one polypeptide to another. The other thing you might notice too is it also goes and can connect one part of one chain to one farther down. So it can also go and attach farther down the chain. Now, as I mentioned before, if we look at our planet, we find that proteins are going to outnumber in diversity carbs, lipids, and nucleic acids. Right? It's been estimated that there is probably 1 billion different proteins out in the world. 1 billion different proteins. Right? That's a lot of different proteins. Right? No wonder that they are the machinery of life, that they run the cells, that they maintain the cells, they repair the cells, right? That they make up the parts of a cell are proteins. So the question that we're interested in is, why all this diversity? What makes them so diverse compared to something like a carbohydrate or a lipid? One of the things, the big points of evidence of showing how living organisms are related to each other by ancestry and descent is by actually seeing that living organisms, regardless of whether it's a bacterium or a fungus or a plant, is that we are all built from exactly the same 20 amino acids. There is a universal pool of 20 different amino acids that are found within living organisms. Now, there are a couple weird bacteria that have 21 and 22, but for the most part, when we look at animals and plants and fungi and algae and protozoans and archaeans and bacteria, we're going to find that every protein is built from the same 20 different amino acids. The same 20. So we have what we call the standard 20. All right. Now we're going to do just a little bit of math, but it's simple math. So you can imagine a chain of amino acids that go and make a protein, right? So, if we have a standard 20, and just to kind of show you the amount of diversity we can get from this, I want to do a little thought experiment. Right. Let's imagine that we have a protein that is really small. It's just based on 3 amino acids. Right? How many different proteins can we build from a sequence of 3? 9. Higher number. More than that. Let me show you. Let's imagine that we have a protein just built on 1 amino acid. Right? How many possible proteins can we have? 20, right? So, if we have a polypeptide that's just made of 2, and each one is 20, how many possible combinations can we come up with? 20 times 20? 400, right? 400 possible combinations. So how much would you get with 3? 20 times 20. You almost need a calculator, don't you? Times 20. 8,000. Just for 3. And the average size for a protein is about 150. Right. That'd be 20. In fact, that'd be 20 raised to the 150th power. Just for a standard protein. That's pretty amazing, isn't it? That's why it's so diverse. When you have a pool of 20 different amino acids, right, you can come up with a huge diversity of proteins with just a few in a chain. So here's a way to represent that. So we have a pool of 20. If you just have 1 amino acid, that's 20 to the first power, that's 20. If you have what we call a dipeptide, 2 amino acids bonded together, how many different combinations? Well, there's 2, and each one is 20, so that's 20 raised to the second power, 8,000. Here's 20 to the 4th power, 160,000. 4 amino acids. 4 amino acids. And if you think about the average size of a protein in our bodies being 150 amino acids long, That would be 20 to the 150th power. And I have a feeling that my calculator may not get that high. It may say error. Let's see if we can do it. All right, 20 raised to the 150th power. Ah, I got the big error. Yeah. Now, I must say that even though it can make all those different possible combinations, not all those combinations are going to be viable. A little later today, we're going to talk about how proteins fold, all right? How they go from a chain of amino acids into a complicated conformation. All right? There are certain rules they have to go by. And even though we might calculate it theoretically how many different combinations, some of those combinations are not going to be biologically viable. They'll just be so unstable that they can't hold themselves together. So, but this is sort of looking at the top point, the highest possible number of combinations. Okay. Now, when you look at the 20 different amino acids, you'll notice that they have one part that is always standard and one part that makes one different from the other. So if you look at what I call the backbone, here's the backbone, you'll notice that every single amino acid has exactly the same backbone. So the backbone is all exactly the same for all 20. What makes one different is the little side chain here. What's in pink? This little side chain is variable from one amino acid to the next. All amino acids are exactly the same except for that little variable, that little side group. And we have a name for that little side group. We call it the R group. We call that the R group. So what makes one amino acid different from every other amino acid is the R group. Now R comes from the word remainder. Right? When you do division and it's the part that's left over, that's the remainder. Well, in chemistry, it's a group that is different, that's variable from one molecule to the next. So that's why chemists call it an R group. It stands for remainder. Okay. Now, this is a very important picture. I'd like you to spend the next couple minutes copying it down, and we'll talk about it. That'll give me a chance to put some holes in our little handout here. Fantastic. Quiz, yeah? Yep. What was that? It is. It's alpha. You're exactly right. Okay, let's talk a little bit about the structure here. Now, every amino acid has the same backbone. What we're looking here is this backbone. We have, to start off, a central carbon. And just like Thomas said, this is more commonly known as the alpha carbon. The alpha carbon. Now, carbon is a really cool atom in that it can bind to 4 different atoms at the same time. It can form 4 covalent bonds. And this is oftentimes why it's at the heart of organic compounds. It can produce a huge diversity by having these 4 different binding sites. If we look here, you'll notice that we just have a hydrogen coming off of it, and here is our R group. That's going to be our variable side chain. Now, you might recognize this functional group. We saw that in the fatty acids. At the ends of the fatty acid, we have this carboxylic acid group, or what we call our carboxyl group. And the reason that we call it an acid is this hydrogen likes to dissociate. It likes to break off and form a hydrogen ion. So this is what makes the amino acid acidic. The other one is a new group. This is called our amino group. Right? And it's simply nitrogen with a couple of hydrogens. Right? So this structure is named for its 2 functional groups, the amino group and the carboxylic acid group, hence the name amino acid. Now, one of the cool things about the 20 different amino acids is that they all have different bonding capabilities. Since their backbones are all exactly the same, it's their R groups that differ, and their R groups can form different types of chemical bonds. We saw a good example with cysteine here, which can form a strong covalent bond called a disulfide bridge. So we have classified these amino acids into 5 major groups. This is all based on their R groups, and this will become important because if we know how these R groups bond, we can predict how the protein is going to fold. All right? So we're going to go through each one of these, and I'll show you why they're in that group and what special characteristic that they give in bonding and in forming the 3-dimensional structure of a protein. Now, just so I don't forget, I made a little packet here that has the amino acids in their respective groups. Here we are. Here we go. They're going to have different R groups. The amino groups are all the same. The only thing that differs is going to be the different R groups. Refers to the R groups. Okay, let's begin with the first page. And here I've put in all what belong to the hydrophobic group. As you can see, there's quite a number of them. Out of the 20, you can see that 7 of them, all right, 7 of them belong to this group. This is a very, very common group. Now, if we look at the name hydrophobic, that means fear of water. Fear of water. Now, when a protein folds, most proteins are going to fold like a ball, all right? Especially if it's something spherical in shape. We call that globular. So most proteins, when they fold, will be in a spherical or globular shape. Right? Let's say that's it. Right? Now, amino acids that are hydrophobic, that have an R group that hates water, right? Where are they going to be located in a protein? Now, think about a protein in a cell. Cell is mostly water. So these proteins are surrounded by water. Where are you going to find the hydrophobic ones? On the inside, on the interior. That's where they're going to be located. They're trying to get away as far as possible from the water. So when the proteins fold, they fold in such a way that the hydrophobic amino acids always end up in the interior. Right. Turns out that's our first rule of protein folding. There's only 4 of them. So that's our first rule. So let me put that up here. So when proteins fold, the very first rule is hydrophobic amino acids will be in the interior. Now, what makes something hydrophobic? Turns out that when you look at a lot of these, you'll notice that the R group is composed mostly of carbon and hydrogen. If you remember the lipids, all right, lipids are generally insoluble in water. They're hydrophobic. And if you look at their structure, something like a fatty acid chain where you'll have nothing but carbons and hydrogens, those are nonpolar molecules. Nonpolar molecules. A common group that we have in those is what's known as a methyl group. We've seen a methyl group in a lot of things before, but all it is simply just a carbon and 3 hydrogens. We call that a methyl group. And it has no interest in water whatsoever. Since it's nonpolar, water has no interest in it. So if you look at our little side chains here, there's a methyl group. There's a whole bunch of just nothing but carbons and hydrogens. We've got 2 methyl groups here. Here we have nothing but carbons and hydrogens. There's a methyl group. There's a methyl group. Nothing here. Nothing but carbons and hydrogens. Here is just a hydrogen. That's probably the simplest one here. But as you can see, when you just have carbon and hydrogen, it's not going to bond with water at all. And that's what makes those hydrophobic. Let me show you another one too. All right. This is called an aromatic ring. All right. And you might be wondering about that, an aromatic ring. Well, what you're looking at here is at every corner is a carbon. Right? And there's usually just coming from it a hydrogen. So let's go and show you what that looks like. So we'll have a little carbon here, a double bond to that carbon. We'll have a little bond to that carbon, a double bond to that carbon, up here to that carbon, double bond to that carbon, and a single bond here. Right? It's a hydrogen. They call it an aromatic because you notice this strange little alternation between double bonds and single bonds. But you'll notice that all there is carbons and hydrogens. And of course, those are nonpolar. So this aromatic ring, as we call it, is going to be hydrophobic. Now, we have a weird one over there. You'll notice tryptophan. Tryptophan has an aromatic ring. You can see it up here. And it has a second one, but we have a little nitrogen there and a little hydrogen. Now, I asked Dr. Cushing about this, and I said, well, that's going to be a polar molecule. Is that going to make any difference? And he said, not really. Even though this one has a nitrogen and a hydrogen, and we would expect a partial, positive and negative charge, this still is hydrophobic enough to be classified as a hydrophobic. So that's a weird one. So tryptophan is a little weird, but still classified as a hydrophobic. The last one is really cool. This one here is called proline, and you'll notice something odd. Every other R group, right, is independent. It all is upright. But you'll notice this strange thing. It's almost like it made a little ring that connected to the backbone. Huh. So this little carbon here, which really should just be by itself, has turned around and gone back on itself and bonded to this amino group. So instead of being nice and straight across, it's kinked. And that turns out to be something kind of fascinating, because oftentimes when you go and you look at a polypeptide when it's folding, all right, every once in a while you'll just see a right-hand turn, and then there'll be another 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 right-hand turn. Every little corner that you see, that's going to be a proline. Prolines cause bends in the polypeptide chain. Right? So anytime you see where all of a sudden the polypeptide chain makes a hairpin turn or makes a right angle turn, you can bet your bottom dollar that there's probably a proline there. There's probably a proline in there somewhere. Weird amino acid. Weird amino acid. Okay. If we have hydrophobic, we also have amino acids that have R groups that love water. And those are going to be hydrophilic. Lovers of water. Okay. Now, let's say that a protein folds and it forms a beautiful little globular shape. Yes? Oh, all these are going to be, these are all hydrophobic. That first page is all hydrophobic. These are all hydrophobic, every single one. Yep, every single one is hydrophobic. Yep. Yep, that first page is just all hydrophobic. That's all you need to know about that. I know in the textbook and stuff they have all kinds of funky little headings, but all you need to know, hydrophobic. Yep, just hydrophobic.
Okay, so the whole pages are separated by the group?
They are. Okay. All the pages are separated by the groups. Yep. All right. So, let's imagine that a protein's folding, and again, it's surrounded by water. Where would the hydrophilic ones be? They'd be on the outside. They'd be on the surface. That's our second rule. So when a protein folds, it folds in such a way that the hydrophilic ones will be on the exterior. Hydrophilic amino acids will be on the exterior. All right. We've only got 2 more to go. That's pretty simple. Now, what makes something hydrophilic? What makes something love water? Well, it turns out there's a couple really simple ones. And the first one I want to show you is this one right here. What do we call that functional group? Hydroxyl group. Remember, hydroxyl groups are found in anything that's soluble in water. Right. Hydroxyl groups, we saw that in sugars. Sugars dissolve in water incredibly easy because they're often loaded with hydroxyl groups. And if you remember, it's because of the unequal sharing. That's a polar molecule there, oxygen and hydrogen. So if you look at serine and threonine, here's a gorgeous one there. A couple hydroxyl groups, those are great. Now, even though tyrosine has an aromatic ring, that screams out, hey, I'm hydrophobic, that hydroxyl group that's attached to it makes it water soluble. So tyrosine also has a beautiful little hydroxyl group. Water makes it hydrophilic. Yep. This is a polar molecule, so water loves that. If you look over here, we've got a little charge here, and it's just the little things balanced. Since this is a what they call a partial charge, it's not a complete charge, so relatively uncharged. Okay. Now these two, asparagine and glutamine, all right, have something that's a little bit odd. All right. We know, for example, that nitrogen and hydrogen actually are polar molecules. All right, that's a polar molecule. So we have a slight positive side over here. When we go and we look at this oxygen and carbon, that's also classified as a polar molecule. We've got a slight negative here. So we actually have 2 parts of the side chain that are going to be attracted to water. Our little amino group here and our little carbon and oxygen. Now, if you go up to histidine, it's this little section right here. This nitrogen and this hydrogen, actually, as you can see, has a charge. And remember, anything that has a charge, water loves it. So water's going to love this histidine. Going to love this histidine. And we also have this strange one. This is methionine. Methionine is something you just kind of have to memorize as hydrophilic. Right? There's probably an unequal sharing of electrons between the sulfur and the carbons here. So we'll probably have a little partial charge around the sulfur. Makes it hydrophilic. But there's something also really important about methionine. Every polypeptide that is made by a cell, and this is any cell. So if we look at the simplest bacterium to the most complicated mammal or fish, or the most towering plant, you're going to find that they all are exactly the same. Every brand new protein that is built by a cell, any type of cell, the very first amino acid in any polypeptide is always going to be methionine. It is always the first one. It is always the first one. Now, it might get cut off a little later, but every raw polypeptide that's made by cells always begin with methionine. It reminds me of I had a wonderful professor at Virginia Tech, Dr. Kosterov. He was a world authority on scale insects, these little mealy bugs that go and sometimes feed on plants and fruits and vegetables. But he and his wife, for every meal or little get-together, or little potluck that we had at Virginia Tech for the entomology department, they were always the first in line. Always. So if we had a potluck, there they were. Always the first in line. All right. If we had a barbecue, they were the first in line. All right. So they were always the first in line. So when I think about methionine, I think about Dr. Kosterov always being the first in line. And we'll see why that is when we get to genes and DNA. Okay. So we've got our two rules for folding. And as you can see, we've done most of the things. We had 7 on the first one, 6 on the second. That gives us 13. All right. So we're getting close to the 20. The next one are what we call acidic ones. And the great thing about that is you'll notice that they have acid in their name. They have acid in their name. That would not be hard. Glutamic acid and aspartic acid. They're also referred to as being negative. And you'll notice the reason they're negative is that, again, they have a negative charge. Right? So glutamic acid, you might notice that this looks like a carboxyl group or carboxylic acid group, that it's lost its hydrogen. All right. So negative. Now, we have 2 negatives. We also have 2 positives. And these are basics. Right? Ones that are basic. You'll notice with these that these have a positive charge. You'll notice we have NH3 here and another little NH2. This is lysine and arginine. Now, the cool thing about this one is that it's our third rule of folding. Right? let's say we have an amino acid that has an acidic R group, and then farther down the chain we have one that's basic. All right? One has a positive charge, one has a negative charge. And what they'll do is they'll go and bond. They'll bond together. All right? Positive with negative. And they form an ionic bond. What do we call a molecule that's formed from ionic bonds? A salt. So sometimes this is called a salt bridge. All right? So, here's our next rule of folding. Acids always bond with bases. Acidic bonds with basic. Acidic bonds with basic. Acidic bonds with basic. So that's pretty cool. Right? So when we go and this thing is folding, it's following some rules. Hydrophobic on the inside, hydrophilic on the outside. Right? As it folds, it goes. In fact, the cool thing, and this is really amazing, the folding doesn't begin after the protein is done. Right? It actually, folding begins as the protein is being made. So it's already started to fold as it's being made, which is really weird. Right? Which means that if you unravel a protein after it's made and then go and have it refold, it'll refold in a different conformation. And that can be a bad thing. We actually sometimes do that on purpose. It's called denaturation. So oftentimes proteins will fold. They'll fold as they're being made. So when they fold, you have the hydrophobic on the inside, the hydrophilic on the outside. All right. The bases and the acids come together and bond. All right, form salt bridges. And we have one more. And that's the one that we've already talked about. Good old cysteine. Cysteine binds with cysteine and forms disulfide bridges. That's our fourth one. So 4 rules. That's it. So if we know those rules, we can figure out, oh, well, that's why they did it that way. And we can also go and start making predictions. Oh, it's going to fold like that. Now, as you get bigger and bigger polypeptides, and it gets hairier and hairier. But it's a really simple way to actually go and come up with something very, very sophisticated. All right. Any questions on those groups and what they do? Okay. When we build a polypeptide, we're doing the same thing that we learned about a couple weeks ago when we talked about dehydration, synthesis, and hydrolysis. If we're going to add an amino acid to a polypeptide, water has to be removed. You can see this. Here, as you can see, we have a hydrogen from the amino group, bonds with a hydroxyl group from the carboxylic acid group. The water drops out, and we get the formation of a covalent bond. Now with proteins, those covalent bonds that join one amino acid to the next along our chain are all called peptide bonds. It's probably a little easier to remember than glycosidic linkage or ester bond. These are just called peptide bonds. Peptide bond, but it's the covalent bond that joins one amino acid to the next in a polypeptide. So this is held together by peptide bonds. Now, if we want to break one off, then we use hydrolysis. Water has to be replaced. All right, oops. I did a little bit more hydrolytic digestion than I hoped to. And we end up getting our monomer back. Okay. Now we've talked a little bit about the rules of protein folding. And to simplify the three-dimensional structure of a protein, we have broken it down into certain levels of organization, certain levels of complexity. And these are just simply numbered. So we have the simplest one, which we call primary structure. Right? The first level of folding we call the secondary structure. Some more folding, which we call the tertiary structure. And then finally, when we have two or more polypeptides that bond together, then we call that the quaternary structure. So let's start off with the first one. This one, as I mentioned, doesn't really exist in real life. As I mentioned before, as a protein is being made, it already starts to fold. So this usually doesn't exist, but for us, this is very useful. We just call this the primary structure. The primary structure. And as you can see, all it's held by is peptide bonds. Peptide bonds. Now I want to show you a couple of cool things here. In fact, I'm going to back up real quick. And talk about the ends. You'll notice that when we take a look at a polypeptide, we have where the locomotive is. This is the front end of a protein, and this is the hind end of a protein. Right? The front end is often where the amino group is. So here's our amino group. So that would be our front. We often describe that as the N-terminal, stands for nitrogen. Right? The hind end is actually where the carboxylic acid group is. Right? And we call that the C end, the C terminus. Right? So we can tell the front from the back, the locomotive from the caboose. So when you look at a primary structure, you can see there's the amino end, that's the N terminus. And at the other end, that's the carboxyl end, or the C terminus. So the front and the back of a protein. I want to tell you a great story. All right. This happened in the 1930s. All right. A world-class chemist working in Pasadena, California. All right. If you watch The Big Bang Theory, you know that's where Sheldon works in Pasadena. Leonard works in Pasadena. All those guys work in Pasadena. You might be wondering, well, where do they work? They work at Caltech. Right? Caltech is a preeminent scientific university in Pasadena. And one of their amazing researchers that worked most of his career there was this gentleman right here. This is Linus Pauling. Linus Pauling is one of the few people to win 2 Nobel Prizes. His first Nobel Prize was on his work in proteins. I'm going to describe what he discovered in just a second. His second Nobel Prize was a Peace Prize. Right? In the 1950s, in the late 1940s, when they were doing atmospheric testing of nuclear weapons, if you went out to, for example, the Nevada desert or the Utah desert, they would explode atom bombs out in the atmosphere. And they didn't think anything of it. All right? But the problem is you have that radioactive fallout that would be caught by the wind and then drift for miles and miles until it rained down on a little desert town. Right? People who lived down from that were often called downwinders. Well, Linus Pauling thought this was a horrible idea, and he started looking to see if there was any evidence that this fallout was causing problems. And he did. He started finding that people in those areas started having high rates of not only things like cancers, thyroid cancer, but also an increase in birth defects. And so what he did is he convinced not only the scientific community, but he also convinced the political community that atmospheric testing of nuclear weapons was dangerous. So they had a ban. They put a ban, a test ban for atmospheric testing. They started testing underground after that. And that's when he won his second Nobel Prize. It was a Peace Prize. Right? But Linus Pauling, as a brilliant chemist, right, figured out this almost universal structure that is found in nearly every protein. And it all goes back to the peptide bond. Now, if we look at the peptide bond between one amino acid and the next, right, so here's our peptide bond. Here's the amino group from one amino acid and the carboxylic acid group on the other. Right. You'll notice something a little bit interesting, is that here's a carbon and here's an oxygen. Those have an unequal sharing of electrons. So the electrons are spending more time around the oxygen. We have a pole. We have a slight negative charge here. If we look at the nitrogen on this side and a hydrogen here, that nitrogen loves the electrons. The electrons love it, so they spend more time around the nitrogen. So the hydrogen has a slight positive charge. So there at the peptide bond, we have a polar region. Slight positive, slight negative. Now what happens is because of that, when you have polar molecules, that can give you the conditions that are perfect for hydrogen bonding. And that's exactly what happens with this structure. So, instead of having a linear thing like this, right, all of a sudden the protein, the polypeptide chain, will go and form a helix. You don't see too many of these anymore. Most everyone's gone mobile. But if you have a landline at home, you might still have one of these. But this is a great representation of what we call a helix. This is an alpha helix. And what actually went and caused this thing to coil is the formation of hydrogen bonds. Again, if you look at what's happening here, here we have, right, our first amino acid. Here we have a slight negative. If you go down to the fourth one, right, we have a slight positive. So we get this hydrogen bond that goes and holds those together. And number 2 will go and form a hydrogen bond with number 5. Number 3 will bond to 6, and so on and so forth. And it forms this beautiful spiral, this delicate coil we call the alpha helix. Yep. It just keeps going down and down and down. Now, of course, there's lots of them. Right? The alpha helix is found in tons and tons of proteins. You saw all the alpha helices in the acetylcholine receptor. Well, here's a common little protein that we find in our mitochondria. This is cytochrome C, and you can see all the alpha helices. This is what we call a secondary structure. Secondary structure. Right. Now, next time, I'll show you the other one that Thomas pointed out when he said, oh, look at the arrows. What do those arrows mean? What you're looking there is the second secondary structure, which we call a pleated sheet, and I'll show you how that works next time. You'll just have to wait. Any questions on what we covered today?